The link between having type A blood and a higher risk for certain stomach cancers is not a statistical quirk. It is a molecular clue. It points directly to the complex sugars, the glycans, that decorate the surface of our cells like a forest of antennae. For individuals with type A blood, one of these specific sugars is N-acetylgalactosamine. This molecule and its presence appears to create a different environment for pathogens like Helicobacter pylori, subtly altering the cellular landscape of the stomach lining over a lifetime. These cell surface sugars are the ABO antigens. They aren't just letters. Type A has the A antigen. Type B has the B antigen. Type AB, logically, has both. And type O individuals lack the final enzymatic step to create either. They are left with the precursor molecule, a simpler sugar chain called the H antigen. Think of it as a foundational structure upon which A and B are built. This single molecular absence, this unfinished biological sentence, has profound and unexpected consequences. Crucially, these antigens aren't confined to your red blood cells. For about 80% of the population, known as secretors, these same A, B, and H antigens are expressed on the surfaces of epithelial cells. These are the cells lining your entire digestive tract, from your mouth to your gut. They line your respiratory system and even your urinary tract. They are the gatekeepers, the very first point of contact for nearly everything you eat, drink, and breathe. This widespread expression turns your body into a landscape of molecular docking sites. Pathogens have evolved proteins that bind to very specific sugars. It is a form of targeted adhesion. A virus or bacterium that develops a surface protein that perfectly fits the lock of a common human cell surface sugar gains a massive evolutionary advantage. It can latch on, invade, and replicate more effectively. Your blood type, therefore, dictates which locks are most prevalent on your cellular gates. Consider norovirus, a common cause of gastroenteritis. Certain strains of this virus have a capsid protein that binds with incredible affinity to the H antigen. This is the antigen abundant in type O individuals. As a result, people with type O blood are often far more susceptible to symptomatic infection from these strains. Their gut lining presents the perfect, welcoming handshake for the virus. Conversely, individuals with type B blood can be relatively protected, as their B antigen structurally masks the underlying H antigen, effectively hiding the docking site from the virus. The influence extends to parasites. The deadliest malaria parasite, Plasmodium falciparum, causes infected red blood cells to express sticky proteins on their surface. These proteins make the cells adhere to uninfected red blood cells, forming dangerous clumps called rosettes. These rosettes can block small blood vessels, leading to severe, often fatal complications. Here, type O provides a significant advantage. The structure of type O cells appears to physically hinder this rosetting process. The clumps are smaller and less stable, giving the immune system a better chance to clear the infection. But this advantage is not universal. What protects you from one threat can expose you to another. The toxin produced by Vibrio cholera, the bacterium responsible for cholera, binds with extremely high affinity to a molecule on intestinal cells whose expression is influenced by blood type. This interaction is strongest in type O individuals, leading to a more severe form of the disease characterized by life-threatening fluid loss. The same blood type that offers a shield against malaria's worst effects becomes a dire vulnerability to cholera. This is the essence of balancing selection. There is no single best blood type. Instead, different environmental pressures from different endemic diseases have favored different types in different parts of the world. A mutation offering resistance to a local plague might also create a vulnerability to a different pathogen. Over millennia, these competing pressures prevent any one blood type from becoming globally dominant. The diversity itself, the presence of A, B, and O in the human gene pool, acts as a species-level firewall. A single pathogen optimized for one type cannot wipe out everyone. So, your blood type is far more than a transfusion requirement. It is an echo of the evolutionary arms race fought by your distant ancestors. It's a signature written in sugar, detailing a biological history of exposure to ancient plagues, parasites, and bacteria. Whether your cells present an A antigen, a B antigen, or the foundational H antigen, is a reflection of a delicate, million-year-old balance between infection and survival. A molecular legacy that continues to subtly influence your interaction with the world today.
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.